everybody. Didn't realize there was a sound component to this video that I'm playing on loop here, so hopefully it's not too distracting. Yeah, so my name's Gianni Castiglione. I started... Can people hear me? Maybe not talking close enough here. Can you hear me now? Yeah, so I'm talking today about owls and whales, the convergent evolution between these two systems, trying to see, how far this rabbit hole goes between these two very divergent lineages. So I'm going to talk a little fast, but I want to get to the punchline at the end. So we're working on the visual system, as many people in this audience are well acquainted with, talking about the photoreceptors in the retina that are actually absorbing light. The way they do this is through these visual pigments called opsins. Opsins are, part of the G protein coupled receptor family, and they have a ligand, more or less, which is this light-sensitive chromophore. It's in the 11 cis conformation. A photon makes it isomerized to all trans. And really, this is the deal with the devil at the heart of vision, because these molecules are toxic. And so today I'll be talking about, it's great to detect light, obviously, it evolves all the time, but all the underlying infrastructure that supports this is very costly, and if anything goes wrong with it, things start to unravel. And so when we're talking about these chromophores that are toxic, they're necessary because what they do is they lead to the conformational change in the protein, and then it's able to signal and something reaches your brain eventually. So the way we classically think about how the opsins evolve is through spectral tuning to different wavelengths of light, and this is most famous, or there's a lot of examples of this in aquatic systems, specifically at depths where light becomes blue shifted because there's attenuation of the light. And so blue light passes most effectively through these layers. And so what you end up seeing is that opsins, rhodopsin specifically, the dim light visual pigment in rods, in species that live in these blue-shifted environments, their wavelength of maximum absorption, lambda max, it shifts to the blue, right? So the deeper you go, the more blue it is, and it's been pretty well demonstrated that this is what's happening. And so a question I've been working on since my PhD, where my supervisor, Belinda Chang, threw a very counterintuitive problem at me, which is, what about things other than light affecting these light-sensitive proteins? Sounds a little weird, but essentially what it is, okay, rhodopsin absorbs a photon of light, as shown here. It leads to a signal, but then behind the scenes, it has to spit out the chromophore. It has to get recycled again so that you can detect light once again. And this simple cartoon is actually extremely complicated. This is maybe 10% of the chemical structures involved. And when we're talking about rhodopsin spitting out light, that just consists of what I've circled here. So rhodopsin spits out the chromophore and then has to be recycled through all these convoluted metabolic pathways, and these are all extremely rate limiting, right? And so it appears that nature has manipulated how quickly rhodopsin will spit this thing out, because it's not able to manipulate how fast these metabolic cycles can recycle things. And so another wrinkle to, another non-spectral or non-photic property of rhodopsin is that it can be activated in the dark through heat, called thermal isomerization or dark noise. We can monitor this using absorption spectroscopy. Over time, you cook these rhodopsins, they spontaneously activate. You can slap kinetic rates on them and see how mutations affect these rates. And they've been optimized for dim light vision. Famous example, people who've known me since grad school are probably laughing because I've shown this slide a million times, where with these toads, you put them in a dark box and they're snapping at flies to see, as a measure of behavior, if they can see. And long story short, the colder that it gets, these cold-blooded organisms can see better. And that's because there's less dark noise. There's less spontaneous thermal activation of rhodopsin. And so you have a better signal-to-noise ratio. And I'm bringing this up because it led to this really weird observation we made a number of years back, where it's well known that this one site in rhodopsin can dictate how much noise there is, this one amino acid residue. And in these transgenic mice, what you can see here, this is electrophysiology, suction pipette recordings. In a wild-type mouse, you see all those little spontaneous jumps in the current. That's dark noise. You make the single mutation, you knock in the this amino acid mutation into the mouse, you get a dramatic decrease in noise, right? Makes sense. Makes sense that all these deep sea fishes have these mutations because they get less noise, they can see better. And so that makes perfect sense. What doesn't make sense is why all tetrapods, to our knowledge, have the crappy residue, which is the one that leads to a lot of noise. And we're talking like whales, owls, bats, things that, like the sperm whale, it needs to be sensitive to dim light, so why would it not figure out the solution that the fishes have figured out? And it's known to limit their dim light sensitivity. And looking at the clinical literature, we realized, okay, well, you know what, there's another thing going on here. It's not just all about detecting light, it is also about the deal with the devil I brought up earlier, which is these chromophores are toxic. And so this mutation, E122, this residue, it makes rhodopsin really stable. It holds on to this toxic thing, gives your body a chance to recycle it. And we know that this is important because in mice, if you mess up, you make certain mutations and you exacerbate this problem of this toxic retinoid accumulating, these mice, you can tell that their retinas start to fall apart. And so the low stability, like in these fishes, right, that's the trade-off. They have high sensitivity, but the protein is unstable. And for them, we don't think it's a problem like it's a problem in other species like mice. We have mice in the lab, and we put them in the dark for a little bit, give them just daylight intensity, and their retina starts to fall apart. But the fishes don't have to deal with this problem, at least these deep sea fishes, because they're physiologically restricted, a lot of them, to certain depths. And so they're able to tolerate this trade-off in a way where they maximize sensitivity, but they don't really care about the toxicity coming from photoactivation. And what I'm showing here is just ranges of irradiance of these different species and the stability of the rhodopsin. And you can see that they start to get really stable rhodopsins when their light environment is very variable. And so we think, tetrapods, they can't tolerate low stability because they're exposed to bright light, a lot of them, especially species that go in between very big extremes. And so we think, rhodopsin's doing something beyond just detecting light. These opsins, they have another moonlighting role where they're trying to mitigate the damage that they actually cause. And if you knock out rhodopsin in mice, they're completely immune to photo damage. They obviously can't see, but they also don't go blind from, we're getting a little twisted here. So what we thought about was, okay, well, if this is happening, it should, there's lots of genes in the retina. There should be other genes that are evolving in these species. And so we looked at a binding partner of rhodopsin called arrestin. And here we're just looking at under dim light conditions, rhodopsin gets hit with the photon, it activates this G protein transducin. But under under bright light conditions, transducin never comes into the picture. Arrestin swoops in there before it ever gets a chance to do anything, and it constitutively binds to it. And so in dim light, in panel B, you have arrestin just turning off the signal, right? Just stop signaling so our vision can be more precise. But under bright light conditions, like all of us right now, arrestin is permanent, it's there for now, and what it does is it binds rhodopsin really stably, and it forces it to suck up all of this all-trans retinal. So it's not just a unidirectional process where it spits out retinal. It's an equilibrium where it sucks it back up. And this protein interacting with rhodopsin, arrestin, it promotes the sequestration process. So it's been long thought that arrestin puts rhodopsin into survival mode during bright light conditions, but it's really hard to prove this because of just the difficulty of keeping mice under certain light conditions that mimic the intricacies of the protein biochemistry. So we looked at these two species that are at high risk of photo damage. Owls, they have diurnal activity, but they have high raw densities, as well as whales, where they have a lot of rhodopsin expression. This puts them at high photo damage risk. It's counterintuitive because they're dim light specialists, but they have to deal with the risk of bright light exposure. What we found is that in owls and whales, specifically using DNDS models, PAML, Clade Model C, we found that whales and owls, they're evolving faster in arrestin. We pinpointed where this is happening in the protein. It happens right in the binding interface of these two things. And we found a mutation, Q69R. Only owls have it, and a couple of these whales, including the sperm whale. We were wondering, what is the effect of all-trans retinal? What is the effect of this mutation on the sequestration phenotype? And this is a bunch of biochemistry, but long story short, we were able to measure the sequestration of all-trans retinal, which means this bar graph summarizes it a little bit better, which is that this single point mutation can increase sequestration of this toxic retinoid by about 30%. We think it's higher because of limitations with our system. And so our model right now is that owls and whales, these are species, there's other ones out there we're trying to investigate as well, where they are dim light specialists, but unlike those deep sea fishes, they have to deal with bright light sometimes. And so how do you balance these two things? Well, luckily you have, a bipartite system here where the rhodopsin can be highly stable, highly sensitive, but the arrestin has evolved an enhanced protective mechanism. So it tells us that, well, if owls and whales that are at heightened risk of photo damage, if natural selection is enhancing this phenotype in them, it's probably at play in other species as well. It's just exaggerated in them. And so we've been doing some interesting things, like we put this mutation into mice, trying to find out if it actually protects them in vivo, as well as trying to package these into gene therapies for mouse models of human blindness, because all transretinal does accumulate over your lifetime and contribute to blindness. So can we steal this solution from evolution and throw it into the clinic? We're going to try at least. So I wanted to just finalize with saying convergence between these two species that are diverged by more than 300 million years really tells us that this is a very important system at play. It's not just all about absorbing light, and that's obviously amazing, but all the complicated infrastructure supporting it can constrain how sensitive your vision can even be. So that's how physiologically costly it is. And so I just wanted to thank my own lab. We're starting out here. We are having some dumplings in Nashville, and my postdoctoral group, and all the work I've been doing with Belinda Chang, and the picture on the bottom, you can tell a little bit younger than the picture on top. My colleague Ryan Schott included. I think he's been aging a little faster. And I just wanted to thank the funding for this and all of your attention. Okay, Rick.
What sort of toxicity, what are the consequences of retention of all trans retinol? In what ways is it toxic to either the cells themselves or to other cells in the retina?
Yeah, so we know if we just give them micromolar volumes, in a cell culture, it kills them. And it does it through multiple pathways, including mitochondria. It also just it stimulates reactive oxygen species. It's a photosensitizer, right? So it can turn oxygen into singlet oxygen. It also forms these condensates with other proteins and lipids in the retina. And in age-related macular degeneration, you get all this garbage that accumulates in your eye called drusen or lipofuscin. And that creates oxidative stress that really upsets the retina, triggers inflammation, and things like that. So there's multiple mechanisms.
And it's seen clinically, as you mentioned, in macular degeneration in people's vision.
Yeah, it's a very, very clinical mechanism. It's just nobody knows what to do about it, because it's not like you can delete rhodopsin, then you'll go blind. So in the clinic now, they have clinical trials where they give people inhibitors of rhodopsin, and so it helps with this problem, but then their vision is way worse at night. And so it's this trade-off they can't get around, and it seems like evolution's maybe done a better job at that. Yeah.
And cones don't experience this because they don't need to be as sensitive?
Yeah, it's a great question. So cones have, in the rods, it's very rate limiting. So you can imagine rhodopsin's like the sink that allows, if the pipe is going out slower in the rods, right, rhodopsin can soak it all up and give the pipe a chance to get all the water out, right? So rhodopsin's a sink for it. In the cones, they have multiple holes, right? They have multiple metabolic pathways that are recycling this stuff so it can go faster. We think it's not much of a problem for them, but we're looking into it currently. Yeah. Thanks.
Okay, hello everyone, my name is Golnar, and today I want to talk about my project on the molecular evolution of turtles' visual opsin in two main groups of turtles, side-neck turtles and hidden-neck turtles. First, I'll start with a little bit of background information, and then I talk about my results of my project. So we know that vision played an important role in the survivability and fitness of many animals that prevail on Earth. It allows for the rapid detection of predators and harmful environments. It also plays a fundamental role in mating behaviors as well as in recognizing prey or nutrient sources. As we see around us, nature provides diverse light environments from the darkness of the ocean depth to the brightness of the deserts. And each animal in this environment has adapted its visual capability to meet different needs. So how do animals see? When light enters the eyes, it is focused on the retina and absorbed by photoreceptor cells in the back of retina. We have two main types of photoreceptor cells, cones and rods, and cones have diverse structures, as you see these four, and rods have higher sensitivity. Typical vertebrate retina, including those of humans, have duplex structures, which has both cones and rods. Vision in all animals ultimately depends on a group of protein, which are called the opsins, and are found within the membrane of these photoreceptor cells. And in a visual pigment, opsins are bound to a light-sensitive chromophore responding to light. There are five main types of photoreceptor cells, five opsin genes expressed in photoreceptor cells, each with different sensitivity to a specific wavelength of light. Rhodopsin, or Rh1, is a photopigment expressed in rod photoreceptor cells, and it contributes to our ability to see in shades of gray, the dotted gray line, in low light environment, when the other four, LWS, SWS1, SWS2, and Rh2, are expressed in cone cells and are responsible for color vision across different spectrums, and having all these cones can give tetrachromatic vision. Visual pigments can change their sensitivity to lights of different wavelengths in two main ways. One is by altering amino acids at spectral sites, which are called spectral tuning sites in the opsin protein. It can also be altered by exchanging one type of chromophore for the another. For example, exchanging A1 type to A2, which is usually associated with freshwater environment, can shift the sensitivity toward longer wavelengths. Additionally, in some vertebrates, including turtles, structures like oil droplets that you can see these ones here, which are in cone cells, act as filters and can limit the type of the lights that reach the opsin. For example, the orange and red can filter out the blue and green light. Testudines, or turtles, are the study group that I work on, which is composed of two main lineages. They diverged around 200 million years ago, and they are called cryptodierans, or hidden neck turtles, and pleurodierans, or side neck turtles. Today, the pleurodierans are less diverse than cryptodierans and are restricted only to southern hemisphere. Members of each group has evolved different neck retractions mechanism. That is their major characteristic. You can see that cryptodierans, they just withdraw their head within the shell, while the pleurodierans just bend their neck sideways under the shell. These turtles, different species of turtles, live in different habitats, from freshwater habitats, land and sea, and each of these habitats have different unique light conditions, leading to varied visual abilities. And this diversity helps these different species of turtles to adapt and thrive in their specific environments. And I should add that pleurodierans are all freshwater species. What we know about turtles' visual system so far, from previous studies, we know that turtles' retina express all five opsin genes, which give them the possibility of tetrachromatic color vision. And several studies on green sea turtles, Chinese, pond turtle, and red-eared slider on the oil droplets of turtles shows that the rods have no oil droplets, when cones have different colors of oil droplets with different numbers. And based on all these studies, all these studies are only on cryptodierans or hidden neck turtle, and we lack information from side neck turtles from their visual. And so for my study, it is predicted that this long divergence time, this 200 million years between these two groups, resulted in considerable differences in the molecular adaptation of their visual opsins. And the objectives of my study was first to see which opsin genes are expressed in pleurodurans and compare them to cryptodurans, and to see if pleurodurans' visual system are under different selective pressures, which is associated with different life stories, and then to review their visual pigment spectral tuning mechanism. To test our hypothesis, first we used 28 whole genome sequence, 34, sorry, whole genome sequences available on GenBank from both groups, and we also used one I transcriptome from one pleurodurans. It's important to have this I transcriptome because we can study which genes are actually expressed in the eye, in the retina. And the coding sequences for all 5 opsin genes were extracted from the turtle's genome and the I transcriptome, and we aligned them using Blasten and Musselin Mega. For the first objective, we saw that all of the opsin genes were expressed in the I transcriptome, and the 5 opsin genes were also recovered in either completely or in part for the majority of our turtle species from both groups. And there was limited evidence of gene loss. For the second objective, to test for direction and strength of natural selection, we used evolutionary analysis model. How they work is that several codon substitution models consider the codon triplets as the unit of evolution, and codon-based likelihood models estimate an omega value, which is the rate of non-synonymous to synonymous substitutions or mutations, that this omega can actually estimate the direction and magnitude of selection on amino acid changes. When omega is equal to 1, it shows the gene is under neutral selection. When it's higher than 1, it shows positive selection. And when it's lower than 1, it shows negative purifying selection acting on the gene. I used random site models implemented in CodeML program of PAML4. And specifically, I used M0 to test for overall selective constraint, and M2A and M8 to test for positively selected sites. In addition, clade models were used to test for divergent selective pressures based on the classification of turtles into their suborders, pleurodurans and cryptodurans, and based on their different habitats, which is marine, freshwater, and terrestrials. For the M8 and M2A didn't show any evidence of positive selection, and our result for M0 model showed that cone opsins are under roughly twice as much constraint as rod opsin, as you can see it here, which is unexpected because rods are usually more conserved due to their role in vision. For the results of our clade model between pleurodurans and cryptodurans, we found significant differences between pleurodurans and cryptodurans in all opsin except for SWS2. The result here showed that RH1 in pleurodurans is positively selected, and the elevated omega in M0 model from the previous slide for all turtles should be originated from the pleurodurans only. And oh, my cursor, okay. In pleurodurans, we can also see that RH2 and LWS is more conserved when SWS1 is less conserved. So as the pleurodurans are all fresh waters, we ran the second one to see if there is a difference, and the RH2 was the only one that showed the difference, which RH2 in fresh waters are more conserved. Next, we studied the variation in opsin genes by aligning our sequences with bovine rhodopsin and looked for substitution at the spectral tuning sites. We found, I just want to talk about the results of SWS1 here. We found 9 substitutions at known spectral tuning sites in SWS1. We know that SWS1 pigment class can be divided into 2 groups. One groups are the UV sensitive ones with lambda max below 390 nanometer, and the other ones are the violet sensitive ones with lambda max between 420 to 440 nanometers. As the results among the substitution, we found these 3 substitutions in cryptodurans and 1 substitution in both cryptodurans and fluoridurans, which were reported to shift the sensitivity towards UV, which is under 390 nanometers. Fluoridurans, all of the fluoridurans had these P93 substitutions that we uncovered these mutations that is inferred to shift sensitivity from UV to violet. And we also found V86 shared by fluoridurans, which has unknown effect. We know that the retina of many animals can perceive UV light, for example, in birds, insects, and turtles. And for turtles, being oriented properly for the hatchlings is a critical survival skill. And even though they've never seen a sea before, they have no problem heading towards the sea. And in this picture, you see the visible, the near UV light is more dominant over the sea, and you see the red and orange light above a city. Studies shows that loggerhead turtle hatchlings exhibit a deliberate avoidance of this yellow and orange lights and show a strong attraction to light within the near ultraviolet to green part of the spectrum. So based on our result, we conclude that first, cone genes are highly conserved over 200, all opsin genes are highly conserved over 200 million years between the 2 groups, and cones are more conserved than rod opsins, which can be due to the role of cones in a specific visual task, such as color discrimination. And also, cryptodurans are predicted to be more UV sensitive than fluoridurans, and building on the result of the third objective, our next project would focus on the spectral sensitivity and evolution of SWS1 in turtles of both groups, and further in lab and behavioral studies are needed to understand the visual capabilities of the fluoridurans and to see what other mechanisms they are using instead of UV light for orientation. I'd like to thank my funding sources and my lab for feedbacks. Thank you.
Hi, my name is Yihang Zhao, and if there's, I'm welcome to any questions. So, yeah, that's the end.
Awesome. Thank you so much for the introduction. Hi, everyone. My name is Taegan. Thank you so much for being here. I'm a PhD candidate at York University in Toronto, and today I'm going to be walking you through some of the research that I have been doing, looking beyond the visual field, identifying shifts in selective pressure on aneurin phototransduction genes across differing photic environments. So aneurins, or frogs and toads, represent the largest group within amphibians, with over 7,000 species currently identified. Unfortunately, they also represent the most threatened group of vertebrates, with about 23% of their species listed as endangered or critically endangered by the IUCN. Okay, perfect. So one of the things that my lab is really interested in is looking at this relationship between the visual system and the environment. And aneurins provide an excellent study system to answer questions about this relationship because they utilize a wide range of habitats with varying levels of specificity across their taxonomic diversity. So as I mentioned, aneurins are found across a wide range of habitats, and that this is important for answering questions regarding the visual system and ecology, but why? So as we know, the environment places selection pressure on genes, and this includes those involved in the visual system. This is due to the fact that all visual detection and discrimination tasks have to take place within the context of the local light environment, and so as a result, visual sensitivity evolves to match that photic or light environment. And this is a pattern that's been established across a wide range of taxonomic groups. So for example, a relationship between water type and visual opsin sensitivity has been established in fish, and this is linked specifically to the different photic environments that exist across water types. So for example, the amount of light and type of visible light available in each environment. But in order to understand this relationship between vision and the environment, we need to have some idea of what's going on under the hood in vision, so to speak. So vertebrate retinas contain several cell layers and cell types, but the photoreceptor cells, which are responsible for absorbing photons of light and triggering the visual process, which we've been talking about a lot today, are the part that we are the most interested in. And there are two types of photoreceptor cells. You have rods and cones. And so when we are examining this relationship between the visual system and the environment, we can look at different aspects of the visual system. So for example, we can focus on those photon-absorbing opsin proteins and their underlying genes, the phototransduction cascade, the electrical signaling component, the visual cycle, which is responsible for retinol regeneration, or even in the non-visual opsins, which are those that are associated with non-visual processes, such as the regulation of circadian rhythm or seasonality. And so in a neuron, some of these visual components, their underlying genes, and this relationship to the environment has been studied. So a relationship between ecology, life history, and spectral sensitivity has been established in opsins and non-visual opsins, in eye size investment and lens transmission patterns, as well as in the phototransduction genes between juveniles and adults in a single species. But one component of the visual system that we were interested in exploring in more detail were these phototransduction genes and this relationship to the environment, but looking at it across a neuron diversity. But what exactly is phototransduction? So the vertebrate phototransduction cascade describes a series of biological steps that convert electrical signals into, or light into electrical signals in the eye. And so this begins with what we call the dark state, which describes the inactivated state of the photoreceptor cell. So at this time, a neurotransmitter called glutamate is being released, which prevents the excitation of neurons and prevents signaling to the brain. But during the light activation phase of phototransduction, the photoreceptor cell is activated by the absorption of a photon of light in the opsin protein, which causes the cell to become hyperpolarized through the closure of these cyclic nucleotide-gated channels, which then produces an electrical signal. At this time, glutamate release is also reduced, and so that allows those signals to reach the brain. But signaling in the photoreceptor is reduced and ultimately terminated during what we call the recovery phase, where the cell is again depolarized, and this again leads to the release of glutamate, and again signaling is prevented to the brain. And so this ultimately returns those cells to the dark state. So this is quite a complex process if you hadn't noticed, and there are about 35 proteins involved in the average vertebrate phototransduction cascade, and each of these is encoded by a gene. Interestingly, within these 35 proteins, there are also distinct copies or isoforms that are found in rod and cone cells specifically. And like other visual genes, those that are underlying the phototransduction cascade are also under pressure from the local light environment across their differing ecologies. And this is something that's been demonstrated in many taxonomic groups. But currently, there is a gap in our understanding of phototransduction genes across a neuron diversity. And so given the importance of phototransduction in the visual system as a whole and the existing relationships that we know for other visual system components and in neuron ecologies, our objective was to explore the molecular evolution and functional adaptations to differing ecologies across a neuron phototransduction genes. And we hypothesized that neurons differing in ecology and life history would possess functional adaptations to their corresponding environments. So we had a couple of predictions as well with this. So we first predicted that a neuron phototransduction genes would be well conserved across their taxonomic diversity, given the functional importance of these genes in the visual system and the overall reliance of neurons on their visual systems. We also predicted that any functional adaptations would be evident through long-term shifts in selective pressure occurring across these neuron phototransduction genes. So to do this, we looked at 123 individuals representing 113 species and 33 families, which is about 61% of the neuron familial diversity. So we looked at this using 91 whole eye transcriptomes and supplemented with 32 genomes available on public repositories. But how exactly do we actually test for this relationship between the environment and visual genes? So we can do this by testing for selection acting on those genes using models of codon evolution. So the Markov model of codon evolution, for example, is able to model biologically important properties of protein coding sequences, such as the ratio of non-synonymous to synonymous substitutions, aka the ratio of changes in protein coding sequences that result in different amino acids over the changes that result in the same amino acids. And so this rate ratio, also referred to as omega, is a measure of selective constraint, where when that value is less than 1, this indicates negative selection, when it's equal to 1, neutral selection, and when it's greater than 1, positive selection. And so these codon-based likelihood models estimate this omega value for us, providing a measure of selective pressure that, depending on its value, allows us to infer the type of selection acting on that gene. And so there are a number of methods that are available to estimate this omega value. You've heard of this one already today. So phylogenetic analysis by maximum likelihood, or PAML, is widely used. So we first used a group of models in this software called random site models that test for variation among sites across a gene alignment. So our first prediction was that a neuron phototransduction genes would be well conserved given their functional importance, and the PAML random site M0 model tests for average selective constraint acting across all codon sites in a gene alignment, which allows us to determine the degree of selective constraint that's acting on our genes. And so what we found was that a neuron the selective constraint on a neuron phototransduction genes was between 0.003 and 0.19. All of those values are well below 1, indicating overall negative purifying selection in these genes, which is something we expected. It's typical in functional genes. This was also in line with the constraint values that we saw in some of the other neuron visual genes, like the opsins and non-visual opsins that some of my colleagues have tested. And while all of these genes had omega values of less than 1, what was also interesting was that we found no statistically significant patterns amongst the types of phototransduction genes. So for example, there were no patterns between those that were found in rods and cones, or those that were involved in activation and recovery phases. But what we do see, hopefully you see it too on the graph, is that there's a lot of variability in selective constraint, which was something we were not expecting. And so we think that this could actually be explained by the variation in ecologies, life histories, and behaviors in neurons. So in order to test this and test for functional adaptation to different environments, we need to first look for any positive change or positive selection, so where those omega values are greater than 1 in our phototransduction genes. And so the PAML M8 random site model tests for positive selection acting across specific codons, or acting at, specific codon sites. And so we did actually find some evidence for a small subset of sites undergoing positive selection across 11 of our 36 phototransduction genes indicated on the figure with an asterisk. But again, we didn't notice any significant patterns between the types of phototransduction genes that we looked at. Oh, there we go. My apologies. Okay, so all of that is great, but how do we actually relate this to the ecologies? How do we look for this ecological correlation to explain what might be driving some of this positive selection that we've seen? So we can do this using models that using analyses that model changes in omega along specific branches or clades. And so PAML provides these for us as well. These clade models test for shifts in selective pressure occurring across different parts of a phylogeny. So what exactly does this look like? So our null model in these tests is essentially just looking at that omega value across our entire phylogeny, whereas our alternative model is that some subset of lineages or taxa have a significantly different omega value, and we typically label these partitions as either a background or a foreground, and we just compare the two omega values to each other to see if they're significantly different. And so we used these clade models to test 7 specific, potentially important ecological partitions across these 11 positively selected genes that we saw. And what these figures are showing here is that for each gene that had a significant result in that ecological partition, what the difference was between the background and the foregrounds that we had selected. Genes that have an asterisk next to them just indicate that for that gene, that partition was the most significant. So for example, in SLC24A1, it was the fully aquatic partition that was the most significant, but there was also some significance for this gene in the fully and semi-aquatic partition and the arboreal. So there's a lot going on here, and I don't have time to talk about all of it, but if you have other questions, I'd be happy to talk about it then. But to give you a bit of an explanation of what could be going on here, let's take a look at the direct developing partition in more detail. So direct developing frogs are those whose embryonic development takes place within their eggs. And so it's been hypothesized that since direct developing neurons do not actually have an aquatic tadpole phase, that there might be different pressures being placed on their visual systems compared to non-direct developing neurons. And so what's also interesting is that we know that some phototransduction genes are actually differentially expressed between tadpole and adult life cycle stages, where one gene is upregulated in the tadpole stage and another is upregulated in the adult stage. And so for these direct developing neurons whose genes are not differentially expressed, potentially because they do not have this aquatic tadpole stage, would this mean we might see some changes in their phototransduction genes? And so that's exactly what we hypothesize is driving these differences in selective pressure in these direct developing neurons. So we believe there also might even be the possibility of a release of selective constraints, so these genes are allowed to change more easily, potentially due to a difference in function or maybe importance compared to these non-direct developing frogs. We can actually test this using an additional model called RELAX, which looks for evidence of the relaxation of selection. And for at least 4 of these genes, we do find evidence for significant relaxation of selection in these direct developing frogs. But we still need to do some additional work to fully assess the functional implications of this. So let's try and sum all of this up. To start, our first prediction was that neuron phototransduction genes would be well conserved, and we did find evidence for all 36 previously documented neuron phototransduction genes, and they were mostly well conserved with omega values of much less than 1, but again, there were no clear patterns between the type of phototransduction genes. Our second prediction was that functional adaptations would be evident through long-term shifts in selection, and we did find evidence for 11 phototransduction genes under positive selection, and these were best explained by the direct developing and aquatic and semi-aquatic partitions. So really what we think is that spending time in water, or how much time you spend in water, has a pretty profound impact on your visual system. So that's all I have for you guys today, but thank you so much to all of our contributors, as well as my lab members, in particular my supervisor, Ryan Schott, and our generous funding sources. If you have any questions or want to get in touch, please feel free to contact me, and I'll take questions if I have time.
So thank you everyone for staying to my talk or coming. So I'm Maxime Policarpo. I'm working at the University of Basel in Switzerland, and I will talk about the evolutionary dynamic of opsin genes in ray fin fishes. So as we have seen in the previous talks, opsins are 7 transmembrane proteins, which will change conformation when receiving light, and this conformational change activates the transduction cascade, which ultimately lead to the emission of an electric signal to the brain. So there are 2 main classes of opsins in vertebrates. Visual opsins, which, as their name indicates, are involved in vision, also called image-forming photoreception, and they are expressed in the eye, so in the retina, in rod and cone cells. On the other hand, there are non-visual opsins, which are involved in non-image-forming photoreception, and which mediate different biological processes, for example, the regulation of the circadian clock or the activation of some DNA repair systems. And that can be expressed in a wide range of different tissues. And contrary to visual opsins, the transduction cascade is much less described and known. So most vertebrates, they have what we call an opsin repertoire, meaning that they have different opsin genes in their genome, each tuned to detect specific wavelengths of the light. Ray fin fishes are particularly interesting when it comes to studying the evolution of this opsin repertoire and of opsin genes, because they live at very different depths, and at those different depths, there are very different wavelengths of the light available. They also live in very different photic conditions. There are some species that live in complete darkness for their whole life, for example, cage fishes. There are species that live in completely turbid water, and on the other hand, you have some species that live their whole life in crystal clear water. So the first goal of my study was to evaluate at what extent does the opsin repertoire vary in ray fin fishes. So to do that, I extracted opsin genes in 535 fish genomes. So I extracted a bit more than 17,000 complete genes, for which you have maximum likelihood phylogeny here on the right. And what you can observe on this phylogeny is that there are clear defined clades, which we call for opsin subfamilies. So in fishes, there are 27 opsin subfamilies, including 5 visual families, so RH1, RH2, LWS, SWS1 and 2, and 22 non-visual subfamilies. We can also represent the data with a species tree here. So this is a species tree of ray fin fishes. And you have 2 bar plots. The first bar plot represents the number of visual opsins, and the second bar plot represents the number of non-visual opsins. And the color of the bar plot depends on the opsin subfamily. So what we observe is that there are great variation in the number of opsins in ray fin fishes. So if we look at visual opsins, the number range from 0 gene in Prietella freatophila, which is a cave fish. So this species lives its whole life in darkness. It has lost eyes, and it is likely that when it colonized cave, there was a relaxation of selective pressure on opsins, leading to the complete disappearance of opsins in this species. On the other hand, at the contrary, in a Tenualosa ilicia, there are 19 genes, which is a maximum found, and this species is known to be highly migratory, meaning that during its lifetime, it is faced with different photic conditions. For non-visual opsin, no, so the minimum was observed in a Pseudoliparis freeway, which is the deepest living vertebrate ever recorded, and which could also indicate that there was a relaxation of selection when this species colonized the deep water. And the maximum is found in the goldfish Carasus auratus with 60 non-visual opsins. But this is also in part due to the fact that this species have a recent whole genome duplication. So as other gene families, opsins evolve by a birth and death process, meaning by recurrent gene duplication and losses. So here on the right, I just put a species tree, which is concatenated at the order level. And you have circles which represent the mean number of genes per species in that order for each of the opsin subfamily. So, for example, what you can observe is that for some subfamilies, the number of genes across the evolution of ray fin fishes is very constant, and there is not much variations. On the other hand, for some other subfamilies, the number of genes is very variable between species, and there are even some cases of complete disappearance of those subfamilies and repeated times during the evolution of ray fin fishes. So to quantify that, I computed the mean birth and death rates for each subfamily, which represent the number of duplication or deaths per gene per million years, and which really indicate the evolutionary rate of those subfamilies. And here is the result. So you have in the y-axis the mean death rate, and the x-axis the mean birth rate. Squares represent visual opsin subfamilies, and circles represent non-visual subfamilies. And what you can observe is that visual opsins are much more dynamic than non-visual opsins with higher birth rates and higher death rates. And this could suggest that there is a higher selective pressure to modulate the visual opsin repertoire than for the non-visual opsin repertoire, meaning that also there is a higher selective pressure on visual visions than on non-image forming photoreception. We then look at the evolution of coding sequences for those opsins. So as we saw before, we computed a DNDS for every gene. So the higher the DNDS is, for here omega, the higher the evolutionary rate of the coding sequence is. So here you have the distribution of DNDS or omega for each gene family. And contrary to the evolutionary rate at the copy number level, we didn't find any difference between the evolutionary rate of coding sequences between visual and non-visual subfamilies. We also saw some nice patterns of evolution between visual and non-visual opsins. So first, we found that there were positive correlation between the number of genes in most subfamilies. We observed also positive correlation between the number of non-visual opsins and the number of visual opsins, meaning that species which tend to have a high number of visual opsins also tend to have a high number of non-visual opsins. And this is true whether you take into account or not species with recent whole genome duplication, which is indicated by those two linear regressions here. And finally, there were also positive correlations, again, between the mean DNDS, so the selective pressure, on non-visual opsins, and the mean DNDS of visual opsins. So what does this mean? It means that there is really a coevolution of the image-forming and non-image forming photoreception in ray fin fishes, and that their evolution are likely driven by the same ecomorphological factors. We can thus wonder which ecological factors. So here I tested the association between different ecological parameters and the number of genes in the different subfamilies, or the total number of visual opsins, total number of non-visual opsins, or even the total number of opsins. So I did that for the copy number, which I will show you, but also with the DNDS, but I will not have the time to show it today. So for the copy number, and for the ecological factors, so we have the depth, the habitat salinity, the climate zone, the gel activity pattern, so whether a fish is nocturnal or diurnal, the diet, the migratory behavior, and the electrogenic abilities, so whether a species can perceive electric signals or not. And here's the result. So you have a tile plot. So each time a tile is colored, it means that there is a significant association, and the color of the tile depends on the R squared. So what we observe is that first, depth is a very good predictor of the number of opsins in many subfamilies, and in most cases, deep living species have much less complete genes than other species. So on the bottom, you have a violin plot, which represents the total number of opsins for different depth categories, and you can see that when you go deeper, you really lose opsins. What you also found is that species with heterogenic abilities tend to have much less genes than other species, and this could suggest maybe there is a sensory trade-off, the species relying more on electric signals to perceive conspecific or predators or prey than vision, knowing that the few opsins that remain on those electric species are under accelerated evolution. I then did a genome-wide scan for co-evolving genes with opsins. So I will not enter into the detail of the method, but basically for each gene in the genome, you can look at if this gene co-evolves with either the number of opsins or with the DNDS, or the selective pressure of opsins. And here is the result. So you have a graph with, in the y-axis, the R squared, which here will indicate the strength of this co-evolution pattern. The x-axis is the p-value, and the red bar is the significance threshold. And each dot is a gene. So each gene that is on the right of this red bar are genes that co-evolve with the opsin repertoire. What we found is that we found some co-evolving genes that are known to be involved in eye diseases when they are knocked out or knocked down in the mice and humans. For example, CAPN15 or CREBL2. And we found significant enrichment of co-evolving genes for light perception genes. For example, crystalline genes, which are involved in the transparency of the eye and the refractive index, or also genes that are involved in the phototransition cascade, such as phosphodiesterases. And we also found significant enrichment for genes involved in the regulation of the circadian clock. Finally, what I did is that I tried to look at where are visual and non-visual opsins expressed, because this is really unexplored, mostly in fishes. So to do that, I downloaded almost 600 publicly available transcriptomes, which represent 14 tissues in 75 species, knowing that I don't have all tissues in all species, so it's not a complete data set. But first, and I only took adult transcriptomes, and I ensured that they were not treated with any chemicals. But to still assess the quality of the publicly available transcriptomes, I first did a PCA using the expression of 6,000 conserved genes. And what I saw is that there is a good clustering of tissue, which is nice because the transcriptome I downloaded have a very different sequencing condition, techniques, and qualities. So we can now go to the result on opsins. So here you have a graphic showing in the y-axis the relative expression, meaning that the expression relative to all the genes of either non-visual opsins in brown or visual opsin in blue, and for each tissue. So without surprise, what you can see is that the expression of visual opsins is the highest in the eye, which is normal. What we are more surprised about is that the eye is also the tissue that expresses the most non-visual opsins. So here. And we saw that in every species, the expression of non-visual opsins in the eye were always dominated by the three same subfamilies, the retinal photoisomerase, RGR, which is normal because this gene is involved in the regeneration of the chromophore, OPN6, and the melanopsin OPN4M2. Then the tissues that also expressed a high number of non-visual opsins were the gametes, so the testes and ovaries. And this supports the recently suggested role of non-visual opsins in the maturation and release of gametes in different vertebrates, but also in sperm thermotaxis in some fish. And finally, the third tissue that expressed the most non-visual opsins were the brain, which also supports the fact that there are some opsins that are known to be expressed in the pineal gland, mainly. So in conclusion, there is a much higher variation in visual and non-visual opsin copy numbers than was than what was previously assessed. There is a faster copy number evolution of visual opsins than non-visual opsins, but there is an equal rate of coding sequence evolution. Ray-fin fish's habitat strongly influence the evolution of visual and non-visual opsins, both at the level of copy number but also at the level of the DNDS. There is really a co-evolutionary network in the genome of opsins and genes that are involved either in the photo-transition cascade or the circadian clock, knowing that I also observe other genes that are co-evolving with opsins, suggesting that they might have a role in light perception or related pathways. And finally, non-visual opsins seems to be highly expressed in the eye, followed by the gametes and the brain. So with that, I would like to thank everyone that was involved in this project. So my PI, Walter Salzberger, Dr. Lily Fogg, and Dr. Fabio Cortesi, all members of the Professor Salzberger group. And I would like also to thank the symposium organizer of the Vision Symposium and everyone here for your attention. Thank you.
Have you thought about taking a closer look at the non-visual opsins expressed in the gonads to see if there's a difference between internal fertilization and external fertilization species?
So one of the problems is that, for example, for the eyes, the expression of non-visual opsins is very consistent across the phylogeny. They always express the same 3 subfamilies. But for the gametes, it's a bit a mess because all the species, they express very different repertoires of opsins. So it's a bit hard to say. And most of the fish, I think they are external fertilization, and the clades that are internal, it's not repeated across the phylogeny enough, I think, to do any kind of test for that. Yeah?
What about your factory genes? Do you see a negative relationship, right?
Yeah. No, there was no correlation between the number of olfactory receptors and non-visual or visual opsins. There is no link at all. Yeah.
For the co-evolutionary angle, do you have any idea, like temporal resolution? Like, do the opsins start to evolve first and the other things have to accommodate it? Like, what's the enabling mutation?
Yeah, that honestly, it's a bit puzzling to me. I don't really know. The thing is that also in this co-evolution pattern analysis, there are some indirect co-evolution. Like for example, most species that don't have a lot of visual opsins, they live in darkness. So in this co-evolution analysis, I will also find genes that may serve the living in darkness, but not necessarily to the eye. Thank you.